So any more? If not, thank you, Priyanka. So we'll be preparing for the next speaker. Before that, the set is ready. Okay, thank you. Thank you. So next speaker is Rupalin Jana.
So, good afternoon to all. Myself Rupalin Jana, doing PhD under the guidance of Dr. Arup Kumar Mukherjee at NRRI, Cuttack. So, today I am going to deliver a talk in my research topic, which is named as Bacterial Endophytes on Wild Rice and their potential for rice plant growth promotion and showing defense response towards rice diseases. Sorry. So these are my contents: introduction, isolation, confrontation, bioassay for antagonistic activities, molecular characterization, greenhouse experiments, plant growth promoting parameters under different field conditions, and conclusion. So these days there is a huge amount of chemical synthetic fertilizers are used for higher yield production and to control different pest management or to protect the plant from different biotic or abiotic stresses. But as we all know, these are very hazardous to our environment also, and they are decreasing the soil fertilities day by day. So instead of these, we can use an alternative approach as toxic free environment, which is termed as endophytic microbes. So as we all know, endophytes are the microbes which reside inside the plant tissues, and they can produce, there is a number of secondary metabolites, and they can help for the total health management of plant crops. But when it comes to our daily routine, means in between the inside experiments to outside experiments, there is a huge gap. So our main aim or main focus to give to stress on the inside experiments to outside experiments, and to find the best solution for the endophyte implication for total health management of rice crops. Again, our topic is endophytes from wild rice. Then, as we all know, or maybe from different researchers, already it has been successfully done that wild rice are the major germplasm resources for rice breeding, and they have the plenty of beneficiary traits, like they can have that drought tolerance and pathogen resistance and high protein content as compared to cultivated rice. So our main aim to isolate the endophytes or bacterial endophytes from wild rice and to implicate the endophytes for total health management of rice crops. So here we choose mainly 5 different wild rice species named as Oryza worthi, Oryza nivara, Oryza rufipogon, Oryza longistaminata, and Oryza granulata. So different plant parts of the wild rice species were collected in ice box and then safely transferred to our laboratory. Then surface sterilization procedure already there is a number of methods, but slight modification we did that 75% ethanol we soak different plant parts in 75% ethanol for 2 minutes, then 3% sodium hypochlorite solution for 3 minutes, and finally surface sterilized with sterile distilled water at least for 5 times. Then the different plant parts cut into different, means at least small parts, the long section in between 1 to 2 centimeter, and then if there is some mistakes in surface sterilization, we can check as plant imprint method, that when final surface sterilization we did, thus from that surface sterilized water 200 microliter we can take in PDA medium or normal TSA medium. Then if any epiphytes was there, they can grow, otherwise our endophyte surface sterilization were successfully done. And long sections where, as we are doing bacterial endophytes, so these long sections were put in triptych Schwager plates and potato texture agar plates. So these are some morphological growth pattern of endophytic bacterial isolates on TSA medium. Then we did some biochemical analysis, that biochemical properties, to see whether the bacteria are producing some biochemical properties or not. So we did gram stain, catalase production, oxidase production, nitrate reductase, urease activity, and citrate utilization. So these are the tabular form of biochemical characterization, where the bacterial endophytes were shown, and gram stain, catalase production and oxidase, nitrate reductase, urease activity, and citrate utilization. So these are some photos of catalase production when our bacterial, one full loop of bacteria were put in when 2% of that hypochlorite solution when put in sorry sir. Sure, okay sir. These are all regular methodology. Okay sir. So we did also confrontation bioassay for antagonistic activities. These are some photos of our confrontation bioassay. So molecular. Sir? Yes sir. Sir, when we did the biocontrol efficacy in vitro condition, then our main total in between total endophytes, there is 3 best endophytes, which is named as Priesta megatherium, which is NRRI EV1, and Priesta aryabhatae NRRI EV2, and Mesobacillus boronifilus, which is NRRI EV3, showed higher quantity of disease inhibition against different pathogens such as Rhizoctonia solani, Sclerosium oryzae, and Xanthomonas oryzae. So we also did molecular characterization using 16S FRNA. So the band pattern in between 500 to 700 base pair. We submitted in NCBI. These are the excision numbers. So in vitro, if the bacterial endophytes is showing many in vitro bacterial in vitro plant growth promoting traits, that's why we did indole acetic production and inorganic phosphate production, and also siderophore production. So here also these 3 endophytes, bacterial endophytes, showed higher amount of indole-acetic acid production, inorganic phosphates production, and siderophore production. So greenhouse experiments also we did in two varieties, TN1 and Tapashwini seeds. So we all know, sir, for treatment of we did seed treatments. Then we put in the greenhouse condition in pots, autoclave soil. Yes, sir. Yes,
Please
sir.
conclude.
So in every cases, so this one the photos, this one is the control one, and these all are the treatment plants. So different plant growth promoting parameters under field conditions also we have seen. Root length, shoot length, fresh root weight, dry weight, and number of yield and yield production also we have seen. So after planting of 45 days, we also give artificially disease inoculation in field condition and in greenhouse experiments also. These are the tables of disease, Rhizoctonia scholani and bacterial leaf blight diseases. So different plant stress related enzymes also we have seen. These are the expression of defense related enzymes. So from all the experiments, we have concluded that mainly three bacterial endophytes, Priestra megatherium, Priestra aryabhatae and Mesobacillus boronifilus were the best endophytes when it compared to higher yield production or antagonistic activities, all activities. So however, the need for further testing in bigger area and different agroclimatic zones also. Thank you. Thank you, sir. Thank you all.
Okay, thank you, Rupalin. So, any pressing questions if you there? Else we'll move to the next speaker.
Rupalin, since this is a this is endophytes, na?
Yes, sir.
As endophytic effect, they won't be individual. There is no possibility of combined effect also.
Yes, sir.
Better you have a inoculum of all mode and also check.
Sir, I can't understand.
No, actually, since you isolated around 8 number of endophytes.
Yes, sir.
There is not only that 8 number of endophytes give individually effects. For example, NRRI.
So, your guide is also here. So, there are a lot of endophytic works earlier in NRRI. So, whether that work has been carried forward or it is there and it is again new one. Dendrophila, it is there. That is fungal endophyte. So that you have skipped. Whether the Dendrophila is not working on sheath blight or other pathogens. That's okay. But as a control, you can also take as a control, because your target is that is sheath blight. Yes. So whether is there any effect on that suppressing for this these target pathogens or not? That you can compare it. Here this may be a bacterial endophytes, but fungal. Salinity. But even when Dr. Dua was there, Madam Dua was telling that it was effective against false spot. Only growth promotion, no antagonistic effect. False spot. Okay.
Matthew was given for false spot.
Hmm.
He has given some 2 endophytes as effective against false spot.
False spot only. Dendrophila was there. Okay. Thank you, Rupalin. Next speaker is Jyotsna Mandal. So please be to the time.
Okay, thank you. So our research in this scenario is to utilize the microbial intervention to revive the soil health status. So if we are concerning about any things, we must need to under the responsible factors for the soil health improvement. These are the main factors, that is minerals, we can say organic factors present in soil, soil structure, fertility, and the climate change obviously, and the soil pH, temperature, and most importantly, as I am working in the soil microflora. And these are the mechanism that is completely related to the three cycle, that is atmospheric cycle, pedospheric cycle, and the hydrospheric cycle. And these three layer is completely linked by the, we can say, the nutrient cycling. What is the biogeochemical cycle? Through this we can link up all the three cycle that can complete itself, and the microbes is the main agent, we can say, to complete that biogeochemical cycle. So for that we have chosen the microbes. And why we have chosen the microbes? Then because soil microbiota, they have different role in the degradation of the organic matter also. Then they can balance the micronutrients and macronutrients present in the soil and make the nutrients available in the soil. And in this way they can improve the soil fertility, stability, and structure. That is why we have chosen the microbes as our primary agent to complete the biogeochemical cycle and maintain the balance of the soil. And I am here today to discuss about one part of the research, that is build a microbial consortium to restore the main nutrients lost in growing paddy. And this is the complete methodology for that part only, that the isolation of the potent microorganism. We have isolated some microbes from the pristine forest area that can complete the cycle, we can say. Then bacterial growth optimization for their lab scale production in the optimized condition. Then application of the microbes and rather the microbial formula to check their effectivity in the crop production. And this is the first part, that isolation. And these are the several microbes we have collected for specific function. We have chosen the 5 nutrients here: NPK, sulfur, and G. These are the microbes we have isolated, and chemical characterization and the molecular characterization has not been completed. Through the process of their effectivity on their nutrient cycling and their effectivity in the nitrogen fixation and potash and phosphorus solubilizer, their effectivity we have checked, but the molecular characterization is now still now pending. So we are going on for this. And next part is the growth optimization for those microbes. That is, one part we have done the optimization of the pH condition in case of lab scale production. So for all the microbes, we have one factor at a time. By the process of one factor at a time, we have optimized the process of the pH. Then we have seen that the optimum condition for all the microbes we have chosen, except the potash solubilizer, except the bacillus strains we have isolated. The main 6 to 7 pH is the optimum pH. We can that's why we have chosen 6.5 for their growth. The optimum pH for all the microbes to grow on in a consortium medium. Next, that is one strain we have completed the carbon source optimization process. There we have chosen the mannitol, and then we have chosen the molasses, that is the industrial waste, and dextrose for their carbon source optimization. And they, again with the different nitrogen source, one we have chosen the high COD that is collected from the industrial waste itself. So there we have seen that the molasses have given, the mannitol have given the highest concentration of the growth of that specific microbes, but molasses, that is an industrial waste, that is showing the similar kind of growth, so we can choose the industrial waste for their optimized production. And the other microbes we have also done, but here I have not seen that the application of the microbial formula to check their effectivity. Here firstly we have chosen the pot study we have conducted based on two crash crop, that is turnip and the spinach. Here from that is the plant height we have measured, that the control sample, there we have nothing we have added in that. In case of test 1 and test 2 is different in case of that their biomass concentration, means the consortium concentration is different in test 1, test 2, but they are improving the growth of the, when we have applied the microbial consortium, the height has been increased. Similarly, in the case of the biomass, means complete from the root to shoot, we have collected, and dry weight we have measured. From that, they have also seen that there has increment in their biomass also. Same we have seen in case of turnip also. Here we have we have added in the microbial formula in a small bed in the paddy field, that is in the control environment, means we have applied in two steps. One is before the sowing of the seeds, and another set was there, that is the 20 days after the sowing of seeds. So there also we have seen the biomass, and the biomass has increased in case of test 1 and test 2 also, plant height and biomass. And in case of the that is when we have after the 20 days of sowing of the paddy, we have seen the similar kind of result, that we have where we have added the consortium. They have seen that the plant height, biomass, as well as plant productivity per sieve, we have, as the area was short, the shorter area, that's why the per sieve we have collected the productivity, that is showing the increment of that also. And these are some chemical analysis we have done, as we have added the NP and K and the zinc and sulfur. We have till now we have shown that sulfur at different time interval, then phosphorus, then nitrogen, they are showing the increment also. And in case of that, that is the where we have added. And from the whole study, means from that part only, we can understand that we have successfully isolated the responsible microbes responsible for the for the isolation or the solidification of the nutrients present in the soil for the completion of the biogeochemical cycle. Then the application of the microbial formula to the paddy and the other cash crop like spinach and turnip gave some promising result. So from that, we can say that the microbial formulation improved the concentration of several nutrients as well as the supporting the healthy plant growth. And these are the references where I have studied some literature. And I would like to thank my funding agency, that is AICT Delhi, and my supervisor, Dr. Ranjita Biswas, and Sukhendu Vera, he is the co-worker with me. He is helping me in the field work. And thank you to the organizing committee and for the exhibition to giving me this chance to present my work here. And thank you to all of you for your patience.
Okay, thank you, Jyotsna. So it was no doubt a nice presentation. So any pressing questions from audience?
Just now I have two questions to you.
Yes, sir.
First question, you are telling it is a consortium.
Yes.
Without identity, how you can tell it is a novel organism?
Sir,
we are optimize the production now.
That I don't want. Just I want without knowing this is a microorganism, which microorganism, because the same microorganism may be identified earlier and it may be used.
Yes, sir.
Then how you can say it is your novel microorganism which is boosting up the...
No, sir. We are not saying these are the novel one.
Then what is
we
the
have
use?
collected the microbes from the pristine forest area. So there we have collected the microbes and isolated in the specific culture media, nitrogen fixing, that phosphorus solubilizer. We have checked their effectivity. So and we have making them together. We are arranging them together so that there is this little amount of consortium can apply.
No.
And
Anyway, what you are telling, okay, but for this type of work, first of all, you should know the identity.
Yes, sir.
That is my suggestion. And second suggestion, unless otherwise, if you check the compatibility, you can't go for consortium.
Yes, we have checked the compatibility.
But there is no information about compatibility studies.
Yes, sir, because of the means I cannot complete.
Because unless otherwise, if you don't know the identity, you cannot have a result. Okay.
We have checked the antagonistic and we have got that promising result that they are not handling each other.
Better have an identity.
So identification is first step that you have to check. Another is you should also have a comparison. In the market there are many consortium are there. Only you have taken your material. But whether your material is superior to that marketed product or not, that also you have to check. If it is inferior to that marketed product, then there is no value. Okay. So, if no question, thank you, Jyotsna. So, next speaker is Prabhanjan Rane.
So another two physical and remaining is 3 is virtual actually. So, yes, sorry.
ମୁଁ ତ ଗଣିଲି 10ଟା।
in the sea and oceans, and only the 2.5%, 2.5% we can say have the fresh water that can be used in agriculture. So out of that, the different fractions are there. So out of that, only the 30% water we can say out of 2.5% water we can use for the agriculture particularly. So now here you can see that what is the global, these are natural resources and what is the global status of our country. So we have the 2.4% land area, 16% human population, 50% animal population, only 4% of water resources. So we have only India have only the 4% of freshwater resources. So that is a very major amount as compared to the other natural resources. So now these are the irrigation statistics of our country. So we have the 4,000 BCM total precipitations, and this is the average data estimated utilizable surface water, groundwater, everything is there. And there is a huge difference between the irrigation potential created and utilized. That is also there. Then this is the irrigation statistics. Now here you can see that this the different sources of irrigations. So here just you can see that the most of the irrigated area comes under the groundwater irrigations. Means the about 60% net irrigated area is irrigated by the groundwater only. That comes under the tube wells and wells, and all the rest 40% area is irrigated by the rest of the surface water, we can say. Now this is the conditions of the groundwater quality, where we have the whole country has the ICAR has studied the whole the groundwater samples almost all the countries, and that found that 45% of the groundwater is poor in quality. What is the poor in quality? Means due to the groundwater have the salinity and sodicity problem. So that is they have also categorized the poor quality water as a saline water, sodic water, that is there. But ultimately we can say that 50% of the groundwater resources we have that are poor in quality. Only the 50% water, about 50% water we have that is a fresh water that we can use irrigation without any adverse effect. Otherwise we have to manage the things. Now the situation is that this is the poor quality water used in the different states. Now the situation is that the per capita water availability is increasing. Now our condition, our country is under water stress condition, and it will be nearly goes to water. It comes under the water scarce country. Now it is the status as the water stress country. The water stress country is that we have the per capita water availability is less than 1700 cubic meter per year. And these are the different lines are there. You can see that these are the some different levels are there. When it will reaches to the lower, then it will country face that type of situations. So now these are the demand of different sectors. And here you can see the water demanded in the irrigation sector, drinking sector, in the industry, energy. In each and every sectors, the demand of water, fresh water is increasing. So we have to, lot of the water is also shifted from the farm to non-farm sectors. That is also a situation. And per capita water availability is decreasing and demand is increasing. And this is the main problem is that the huge quantity of fresh water due to a rapid urbanization or the improving living conditions, the huge fresh water is diverted to the by using the peoples, and that a huge quantity of wastewater generated. So the situation is there, one side we have the scarcity of water, fresh water for agriculture, and another the situation is the disposal of wastewater. We have to, huge quantity of wastewater we have, where we have to dispose it with a safe and sustainable manner. So that we have planned that we have to use this water, only the wastewater is having the problem of pathogens and metals condition. Otherwise it have the positive benefits, positive negative things are there. Means it has the merits and demerits. So merits, it has a nutrient rich water, easily alternate, low cost water is there, availability is there. So these are the positive things. But and side by the negative things are that it has the pathogen loads, metal content that can harm the soil health also, the plant health also, and the consumer health also. So that is there. So we have to use that water in a judicious and a sustainable and safe manner, so that we can avoid the food chain contamination. So we cannot go for this wastewater irrigation for food crops. So we have to use this water for the non-food crops. So non-food crops may be flower crops or maybe turf grass. So that we have used this water for turf grass cultivation or turf grass maintenance. So that we can, whether the turf grass will be feasible or that we can alternate a safe and sustainable source is there, or it is feasible, and what will be the happen when we use this wastewater for turf grass cultivation. No, so now another many arguments are there that we have why we are using this water for turf grass, because turf grass is require more nutrient or continuous water it needs, it grows near to the peri-urban areas. There are different things are there. The different factors are. That's why we can go the wastewater irrigation for turf grass. So now that this we have conducted one experiment, what is the turf grass performance under the wastewater irrigation when we have planted the turf grass in two planting methods. One we have taken the subsoil surface mulch and another is normal planting, and we have taken the three irrigation scheduling criteria that is based on the 75% ETC, 100% ETC and 125% ETC. So now this is the quality of the water that we have used for the irrigation or the experimental crops. The groundwater quality is okay, that's no problem. As all parameters were within the permission limit. The wastewater has some high organic pathogen loads and the BOD content was more than the permission limit. Otherwise, all parameters were safe as per the maximum concentration or the irrigation standards are there. Now, these the meteorological parameters we have collected, through which we have calculated the ETC, daily ETC basis we have collected, and the weekly irrigation we have given, cumulative. On the basis of the cumulative ETC data, we have given we have calculated how much water we have applied for a particular plot that we have calculated, and this one digital flow meter is there. Then we have exactly the water that needed by the plant for particular ETC that we have applied for a particular plot. Now this is the meteorological program, and here you can see the photographs of the turf grass conditions by under the experimental. And now this is the crop water requirement. The ETC we have calculated through the crop water model also, and we got the total ETC water requirement. We can say the actual water requirement calculated ETC of turf we have calculated using the crop water model. So we got 915.7 millimeter as a crop water requirement of the turf grass. And then another using the soil water balance method, we have also calculated this is the different input parameters on using this input parameter, and by using the formula of soil water balance method, we have calculated the crop water requirement of turf grass, and then we have found that this much the range here you can see. This is in the last year here is range from I think it goes to 927 to 1215 cm is there. So then we have also calculated we observed the rating quality on visual observation basis, the quality of the turf grass, and then we have considered the quality ratings with the best quality rating which considered the crop water requirement that we have chosen. This is the best one. So according to that, considering the quality rating of the turf grass and the water requirement, we found that this is the 920 centimeter of this is the best one, that is water requirement of the turf grass. And we have found the treatment that we have observed 125% ETC at subsurface soil mulch. That is not that we have the treatment we have. That where we have given the irrigation at 125% ETC, and the planting method was the subsurface soil mulch was there. The quality of turf was as compared in the sense of color or texture and many visual observations on traffic tolerance, many parameters we have observed. On that basis, we have found that this is the optimum water requirement is 920 centimeter of the turf grass, and the irrigation scheduling criteria is 125% ETC. So that is thing, and these are our authority directors, project directors that we have provided the facilities to conduct these experiments, and we have published one paper also on this turf grass. And this is thank you very much.
Thank you, Dr. Gujjar. So no doubt it is a very good piece of work. So we invite some questions from audience. Any question?
Only one question,
Yes.
two question.
Yes.
So actually, turf grass it is not we are going for any commercial or we are not going for any production aspect.
Yes.
Probably we will be growing it as an aesthetic value.
Aesthetic value and for decoration or for
So how far this usage of wastewater will not have impact on ours, because mostly we will be using if you see the turf grass, we used to roll this thing, that thing.
Yeah, that's we have mixed conditions. When we are go for irrigation, after 4 or 5 days, we cannot allow the public to move in the cover.
Like pesticide also, you are saying there should be some waiting.
That conditions we have put. After 4 or 5 days, after irrigation, we have to stop the public movement for 4 or 5 days. After that, we can do the crop.
Okay, sir. Why not we have some pre-processing also, sir? Is there any possibility?
Yeah,
for that?
it's there. That pre-processing is there, okay. But the condition is that when it is partly, mostly occurs in there. Many things are there where we cannot move in the rough grass, but we just maintaining the things of the funds, everything that we have to maintain. So that purpose we can use this water.
So this was undertaken from 2013 to 16. So after that, any further study has been done?
No, sir. Another scientist, one scientist is there in the Museum of Agriculture and is doing that particular area.
So have you developed any recommendation in terms of any bulletins? Yeah, yes, you have your.
Based on wastewater irrigation, but general irrigation. Even we have contacted with the BCCI, the Board of Irrigation Control of India. The team have reached IRI, they have seen classes. And we have done many classes are there. They have selected some classes for their cricket match or cricket. So that is also there.
Okay, if no questions, we thank Dr. Gujjar for nice presentation. So next speaker is last Swagatika Das.
Another 4 is there. Including you, 4 is there. So let us return to that.
Good afternoon, respected sir and other delegate members. I am Swagatika Das. Currently I am working as a PhD student under the guidance of Dr. Lambodar Jaina, and I am going to give a topic. I am going to give a talk about the topic is.
Use microphone.
My topic is assessment of phenotypic variability under normal light and low light condition in diverse rice genotypes. Let's come to introduction slide. On the introduction slide, low light is a major constraint in rice production because of insufficient light due to cloud cover during Kharif season in the eastern and northeastern regions of India. It affects all stages of rice, but more at the reproductive stage. It reduces tiller number, grain number, panicle number, grain weight, and grain fertility, and ultimately it reducing grain yield approximately 35%. The core of crop improvement is phenotypic diversity, which makes it possible to identify super cultivars that are appropriate for different rice varieties in both normal and low light conditions. These are my objectives. First objective is to assess the phenotypic diversity of rice genotypes both under normal and low light conditions. Second objective is to identify the donors for all major traits related to grain yield under low light stress conditions. On the methodology slide, first step is selection of diverse rice genotype. A total of 192 diverse rice genotypes are chosen for phenotypic evaluation for grain yield and related state under low light condition. This comprises of 123 lines of lantaceous and high yielding varieties, 60 lines from millet irribreeding lines, and 9 popular rice varieties. These are my phenotypic design, phenotypic evaluation of diverse rice genotypes. There are total 192 number of genotypes. My topic deals with low light stress, so light treatment is done. Normal light is 100% sunlight is available to crop area, but but using agro shade net there is a low light treatment condition. Under Rabi season there is 50% light cutoff, and under Kharif season there is 25% light cutoff. Our design is alpha lattice design with maintaining 2 replication. I am taking two seasons, that is two Kharif season, that is 2022, and Rabi season 2023. Phenotypic evaluation of traits. Observation were recorded on 5 plants from each replication under both NL and low light condition. These are my traits. These are total 13 traits. Test to 50% flowering, plant height, tiller number, panicle length, grain number, spikelet number, spikelet fertility percentage, panicle weight, 1000 grain weight, biomass, and harvest index. These are all related to grain weight. These are under adjust light. This is the field condition. This is 100% normal light condition, and this is low light condition using agro shade net. This is vegetative stage and this is reproductive stage. This is Kharif 2022 and Rabi 2023. A collection of 192 diverse size genotypes are phenotypically evaluated under open shade, under open condition and shaded condition. All the 13 traits show significant difference between normal light and low light, but except test to 50% flowering. This table showing descriptive statistics for grain-related related traits for association mapping panel genotype under normal light and low light condition during Kharif season. These are my traits and these are their range, respective range under normal light and low light. Except plant height, all traits are showing negatively reduced, but only plant height is showing positively changed. All traits are showing significant reduction. For under Rabi, this table shows descriptive statistics for grain of Rabi 2023 season. Except DFF traits, all traits are showing significant reduction. The normal distribution graph of all agronomic traits. These are both under normal light and low light. All traits are showing normally distributed. 3-way ANOVA diverse rice genotype for granular and related trait. 3-way ANOVA means there is 3 conditions: light condition means low light and normal light, genotype, how many genotypes, and seasons, that is Rabi and Kharif season. Except light, under light condition, except DFF, all traits are showing significant, but between light and genotype and season, all traits are showing significant variance. This graph showing the frequency distribution, all significantly distributed traits under low light condition exhibit variation compared to normal light condition for both the Kharif and Rabi season. This graph showing the difference between low light and normal light between each traits. This is the correlation plot. The correlation plot displays the Pearson correlation coefficient assessed by p-value. In both the Kharif and Rabi season, grain yield exhibited significant correlation with tiller number, spikelet number, grain number, biomass, and 1000 grain weight. In the figure, slight yellow cells with slight yellow color showing that positively correlated with those traits. This graph showing grain yield dependent PC under normal light and low light. Under this season is Kharif 2022. Grain yield dependent PCA plot showing fast PC1, PC2, PC3, PC4 comprises total 74% of the variance under normal light and 73% under low light condition. And under Rabi 2023, there is under normal light condition, that is 71%, and low light is 72%. Under conclusion, this comes to conclusion. This study demonstrated wide phenotypic variation making suitable for GWAS analysis. Means low light mainly affects the eastern and northeast region of India. So my study mainly mainly give importance to those rice variety, mainly NRRI rice variety and NEH center and BCTD center. So genotype exhibited better performance for grain and its related traits on the allele condition like Sonam-Prabha, Purnendu, TRB 47, TRB 68, Magan BCTD 62. Sonam-Prabha is low light resistant variety, and these all variety are showing resistance to low light condition. This information is valuable for breeding programs aiming to develop low light stress tolerant rice varieties. This panel would be used for GWAS analysis to identify mining superior haplotypes or allele for different traits. Thank you.
Okay. So, Swagatika, thank you. Any question from audience?
So, there are two questions. One question, whether these varieties behave in the same manner under both seasons, or is there any difference? Because Kharif Rabi there is a difference with light also. So I'm asking whether these varieties behave in the same manner in both the seasons. Ah. And second thing, already reported genes are there for low light stresses, na? Already reported genes are allele. Whether you checked, is there any report of these genes in this?
In that low light stress is not identified yet.
No gene, single gene. Okay, now you are going to do GWAS, based on that you will be identifying the genes.
Okay, Swagatika. These are already Purnendu, Swarnaprabha, already it has been reported earlier. So again, other lines, varieties. So these varieties from here. These are lines, breeding lines. Okay. So this is from Dr. Lambodara. Go to the previous slide. So basically, is from our line. So only these two. TRB 467, 468. These are genotypes, na? Not Swarnaprabha, Purnendu, they are varieties. Okay, thank you.
I think TRB 4.
They are lines, na? Okay. So now we have completed the physical presentation. So we'll be switching to the online presentations. ऑनलाइन है कोई? Are they online?
Yes, the first will be by Prabhanjan Rane.
Okay. So, Mr. Prabhanjan Rane, so please share your presentation.
Prabhanjan, you share your screen.
Sir, my screen is visible.
No, not yet. Yes,
Now, sir.
now it is visible.
Prabhanjan, the time is 8 minutes for your presentation and 2 minutes for discussion. Please stick on to the time.
Okay. Can I start, sir?
Prabhanjan, do you hear me?
Yes, you are Please start your presentation.
Time is 8 minutes for your presentation and 2 minutes for discussion. Please stick on to the time.
Okay. Good evening, everyone. I am Prabhanjan Rane from Division of Fruits and Horticultural Technology, Indian Agricultural Research Institute, New Delhi. Now I am going to present an oral presentation on the topic palynological studies of different grape species for ascertaining their utilization in grape rootstock breeding. India is a major exporter of grape. In 2022 to 23, India has exported 2,67,950 metric tons of fresh grapes, which is valued about $313 US million. Grape cultivars with a pollen germination rate equal to or greater than 30% can be employed as an effective pollinator. In grapes, pollen plays a critical role in grape production, and its pollen germination potential is a main factor. Palynology is the study of pollen grains, and applied part of the palynology can be used for taxonomic classification, pollen storage, and compatibility studies among different species of the genus. Also, in hybridization program, asynchronous flowering poses some challenges which can be overcome by the pollen storage. And in case of grape, popular grape rootstocks have a narrow genetic base, which are derived from few American grape species. Also, Vitis parviflora, it is a Himalayan wild grape species, possesses a moderate vigor, drought resistance, multiple disease resistance. It is a good parent for grape rootstock breeding. So, in this perspective, research limited research has been conducted on the palynological aspects of Vitis rootstocks, and there is untapped potential in the exploitation of indigenous Vitis species for rootstock breeding. In this perspective, objective of my study was to study the pollen ultrastructure and storability of grape rootstocks, and to analyze cross-compatibility of Vitis parviflora with other Vitis species. In plant material, we have used three genotypes from three different species. And we have checked the cross compatibility of Vitis parviflora with Vitis vinifera cultivar Pusana urang and Vitis champigny cultivar salt creek. For ultrastructural studies, we have followed the pollen collection method as follows. We have bagged the inflorescence before the opening of flowers, and they were harvested at the 50% flowering stage, and these were dried for 24 hours at room temperature. Then these separate pollen grains were separated by sieving, and such pollen grains are stored in a small vials in 3 to 5 degrees Celsius in a silica gel desiccator. And the scanning electron microscopy was performed according to protocol given by Nikolic and Milatovic. For pollen storage studies, 4 treatments were studied, like room temperature 4 degrees Celsius, minus 20 degrees Celsius, and minus 190 degrees Celsius, which is the storage of pollen in liquid nitrogen. And the pollen germinability was observed after first day, third day, fifth day, seventh day, and 50th day and 30th day of storage. In vitro pollen germination was studied on the media 10% sucrose plus 100 milligram per liter boric acid and 300 milligram per liter calcium nitrate, which is the standardized media for grape pollen germination. In cross-compatibility study, pollen tube growth, pistils were sectioned using a freezing microtomy, and their pollen tube growth was observed by aniline blue fluorescence microscopy. And the various compatibility parameter, compatibility index parameter, cross-compatibility parameters like berry set percentage and average number of seeds per berry and compatibility index was calculated. And this whole experiment was carried out at a Division of Fruit Science, Indian Agricultural Research Institute. Research and discussion. The flowers of the Vitis vinifera cultivar Pusana urang was hermaphrodite with well-developed pistil and stamens. In scanning electron microscopy, we have found that the pollens of Pusana urang was tricolporate. Pollens of Pusana urang were tricolporate with foveolate perforate exine ornamentation. And the polar axis was 26.09 micrometer and equatorial axis was 14.78 micrometer with PE ratio with 1.76, which categorized this pollen into prolate shape. In our Vitis champigny cultivar Salt Creek, male flowers was there and the rudimentary ovary was present, and but the stamens were well developed. And the scanning electron microscopy of the pollen grains of this cultivar revealed the pollen grains were tricolporate with presence of 3 furrows. with the foveolate perforate exine ornamentation. But the polar axis was a 28.37 micrometer and equatorial axis was a 15.03 micrometer with P ratio 1.8, which categorized this pollen into prolate shape. In Vitis perviflora, the semi-reflex stamens were observed, and the pollens were inaperturate with absence of aperture. This makes them polarized circular or sunken regions were found in these pollen grains. And the exine ornamentation of this genotype was foveolate perfolate, but the number of aperture aperture was not present, and the polar axis was a 23.52 micrometer and equatorial axis 18.75 micrometer with a P ratio 1.25, which categorize the pollens of this genotype into subprolate shape. The effect of different storage temperature on duration on in vitro pollen germination of Vitis vinifera cultivar Pusa naurum. In this genotype, we have found that the highest germination was recorded at D0, which is immediately after harvest, which is 61.3%.
थोड़ा रुक जाइए सर, थोड़ा ये थोड़ा सिस्टम के इशू के कारण आपका जो है स्क्रीन जो स्ट्रीम नहीं हो पा रहा है, लाइन पे बनने से थोड़ा.
Okay. हेलो.
जी सर, लाइन पे बनना है थोड़ा सर, सिस्टम में इशू आ रहा है.
Okay.
तो आपका जो है पीपीटी को स्ट्रीम किया नहीं जा पा रहा है.
हेलो
सर लाइन पे बनना है थोड़ा सा, सिस्टम का जो है वो जूम का इशू हुआ है। जी सर। सर मैं आपको बोलूँगा जब सही हो जाएगा, वेट कीजिए थोड़ा सा, 5 मिनट वेट कीजिए।
ठीक है।
सर एक स्टार्ट ना।
You can continue. You can continue.
Okay. The effect of different storage temperature and duration on in vitro pollen germination of Vitis vinifera cultivar Cuttack. Here we have found that the pollen germination at D0, which is at fresh pollen germination, which is highest in all storage conditions, and pollen stored at minus 196 degrees Celsius and minus 20 degrees Celsius recorded. Pollen stored at minus 196 degrees Celsius and minus 20 degrees Celsius did not show any significant difference in pollen germination rate from initial pollen germination up to 30 days of storage. But at room temperature, pollen germination on third day of pollen storage fall down to 16.3 percentage. At 4 degrees Celsius, pollen can be stored up to D7, which is day 7, 7 D7, without significant loss in initial germination percentage. In case of Salt Creek cultivar, highest pollen germination was recorded on D0, which is 76.7%, and minus pollen stored at minus 196 degrees Celsius and minus 20 degrees Celsius recorded significantly higher germination rate without any loss in pollen germination up to 30th day of storage. But pollen stored at room temperature showed only 46.7% germination rate on 7th day of storage. This is the picture showing variation in pollen germination rate of pollen stored at different storage condition up to 30 days of storage. Inaperturate pollen in Vitis parviflora did not show any pollen germination. So pollens here we cannot found any pollen tube growth. This is the results of cross compatibility study. Here we have found this from this image we can say that the in flowers of Vitis parviflora crossed with Pusa Pusanauram. We can see here pollen tube penetrating inside the ovule, but in Parviflora and Salt Creek, pollen tubes reach the base of the base of the ovary. So these two combination can be compatible with each other, but in cell flowers of Vitis parviflora, we have not found any pollen tube growth, although the pollen sufficient quantity of pollen were deposited on the stigma. Fruit setting rate, average number of seeds per variant, compatibility index of two interspecific crosses shows that highest berry retention was in Vitis floriflora cross with Vitis champini, Salt Creek, and lowest in Vitis floriflora cross with Vitis vinifera pusanaurum, which is 52.05%. And compatibility index in VP cross with VC was 1.183, and VP cross with VV was 1.154. Conclusion. Vitis flora displayed inaperturate pollen which didn't germinate in vitro. Hello. Hello.
Yes, you are audible.
My screen is moving.
Yes, Prabhanjan, you are audible.
Yeah.
I am on conclusion slide. Vitis flora displayed inaperturate
You are
pollen.
audible.
Which didn't germinate in vitro, while Vitis vinifera pusanaurum and Vitis champini cultivar Salt Creek exhibited tricolporated pollen with successful in vitro pollen germination, and effective pollen storage conditions were identified at -20 degrees Celsius and -196 degrees Celsius, providing a practical solution for addressing challenges related to asynchronous flowering during pollination. Cross compatibility studies indicated high compatibility between Vitis parviflora and Vitis vinifera cultivar pusanaurum, as well as Vitis parviflora and Vitis champini cultivar Salt Creek, suggesting their potential use in grape rootstock breeding. Notably, inaperturate pollen and incapability of setting berries of self-flowers in Vitis parviflora reveal its male sterile nature, which ensures elimination of tedious and laborious emasculation procedure during hybridization. So we can go for hybridization of Vitis floriflora without emasculation. So as part of this is the part of my research article titled "Studies on Pollen Micromorphology, Pollen Storage Method, and Cross Compatibility Among the Grape Genotype," which is accepted in a Frontiers in Plant Science. Thank you.
Okay, thank you. Prabhanjan Rane. Any question?
Yes, sir. हेलो
जी सर, लाइन पे बना सर, फिर से वही इशू हुआ है सर।
ठीक
sir. Sir, నేను coming, sir. నేను next week main sir. I'm not taking watch. Yes, sir. Next week main sir. Sure. Next week my presentation only, sir. I'm not. Voice, sir. Voice. From the external, I'm not. Actually, next week my presentation only, sir. But now it would be see the host only, sir. Host is logged out, I think, sir. Okay, sir. Okay.
Hello. Prajan, thank you very much.
Thank you, sir.
Next, we will move on to Kishore. Ankala Sai Kishore. Ankala Sai Kishore, are you in line?
Sir, am I audible, sir?
Yeah. Please share your screen and start presenting. Please stick on to the time.
Yes, sir.
You are given 8 minutes for presentation and 2 minutes for discussion. Please.
Can you see me? Sir, can you see my presentation, sir?
Yeah, your presentation is shared. Just you move to check it. You move slide to check it. You move one slide to check it.
Yes, sir. I have shared my presentation, sir.
Yeah. You shared. It is visible for us.
So next slide, you just move to the next slide. It's not moving.
Then unshare and share once again. Please unshare and share once again.
Sir.
Yes.
Okay, now you start.
It is moving.
Yes, sir. Yes, sir.
Okay.
Dr.
Start your
Sir,
presentation.
my name is Sai Kishore, sir, and I am here to present the topic on the impact of legume residues and nitrogen levels on soil biological properties, sir. So mainly in this condition, sir, due to cell system, sir.
Sai
So we are
Kishore,
known to
your
add
voice
upon
is
it,
not audible and clear.
sir.
Your voice is not audible and clear. Okay, you continue.
Now, sir, is it audible, sir?
Okay, you continue.
Yes, sir. Due to cell cropping systems, sir, due to cell cropping systems, there has been high amount of impact on the soil properties, sir. So thereby we have changed it to the legume-based cropping system, sir. So with adding the legumes, there has been a increase in the yields also, sir. Increase in the yields, thereby increase in the soil properties also, sir. That can be soil dynamics, sir. Mainly focusing on the bacteria, sir. So bacterial, fungal, and even this biomass, carbon, and nitrogen, sir. So based upon all these parameters, sir, we are mainly focused on to increasing the legume-based system with the above the plant parts and below the soil in the micro parts, sir. So that will enhance the soil properties. So thereby we are taking the objective to study the effect of this legume residues on the soil bacterial population, and even on the increasing the soil CN ratio, biomass, carbon, and the nitrogen in case of rabi maize, sir. These are my objectives in case of my article, sir. So the main motive is to check the legume based crop system, sir. In this we have taken the soybean, green gram, and groundnut, sir. In which they have been taken as a kharif crop, sir. Followed by the, in case of rabi, we have taken the rabi maize, sir, with main plots are 6 and the subplots are 3, sir. Mainly to check the whichever cropping system that is best suitable in case of southern, followed by subplots, sir. In case of six main ribbon, we have taken the groundnut, soybean, green gram, sir, with 100%, 75% RDN. Followed by the in case of subplots, we have taken 100, 125, and 150% RDN, sir. The mainly focus on to check the bacterial population count, sir. Here we have taken the bacterial count with a serial dilution, followed by biomass and carbon, nitrogen, with the fumigation and the fungation method, sir. To estimate the total carbon content was taken in case of ignition method, sir. And thereby we have taken the nitrogen also, sir, with the similar parameters only. As you can see here, we are taking the groundnut, soybean, and green gram, sir, followed by 100, 125, 150% RDN, sir. We need to check whichever cropping system is mainly suitable for the rabi maize preceding groundnut, sir, or soybean or green gram, sir. So here comes my results, sir. Mainly check on this effect of these legumes on the rabi maize, sir. We can see here, sir, green gram with adding 75, 100% RDN show its higher, sir, followed by the green gram 75%, sir. We can see 100% RDN, we show its higher values in case of biomass, carbon, followed by this 75%, sir. Similarly, in case of rabi maize also, sir, 150% RDN showed its superiority, sir, followed by 125, 100% in the biomass total bacterial count, sir. So when it comes to biomass carbon content, sir, as you can see, higher amount of biomass carbon content was seen with the green gram 100%, followed by the green gram 75%, sir. This was higher in case of 100%, followed by soybean and groundnut, sir. The subsequently was seen in case of 150%, followed by 125 and 100% RDN, sir. The similar trend was seen in case of biomass nitrogen also, sir. This could enhance the nitrogen activity, sir, mainly in case of 100% RDN, sir. This 100% RDN showed higher values due to high amount of biomass generation. This biomass generation showed significantly increased values compared with the 75 soybean and groundnut, sir. So similarly, in case of rabi maize also, sir, 150 showed higher values, sir. This is due to high amount of biomass generation, even the higher amount of yield production, sir. When it comes to the CN ratio, sir, we can see high amount of CN values in case of green gram 100 to high amount of green gram residues, and even the high amount of times contents are also enhanced, sir, in case of green gram with 100% RDN, followed by the soybean and groundnut, sir. The similar fashion was seen with the CN ratios also, sir. We can see high amount of CN ratio, sir. There is about around 5% is seen in case of green gram, sir. Subsequently, lowest was seen in case of groundnut, sir. And the last comes the humus fraction, sir. We mainly looked into the how much amount of humus fractions were seen in case of 170%, sir. You can see a amount of seen in case of 100%, sir, followed by the 70%. But however, humus fractions are not much changed due to there have been a long-lasting effect. So though they are not much influenced, but they are shown as a slightly positive note in case of 1.5 year of study, sir. So based upon all these parameters, sir, so in case of soil bacterial count also, sir, you can see a increasing trend at days of showing, sir. This is mainly due to the residues that we applied in case of Kharif that has a positive influence in case of rough maize soil, sir. This trend was seen in case of 30 days with an increasing trend, or subsequently decreasing in case of 40 and 60 days, sir. This same trend was seen in 2 years of study also, sir. When it comes to the urea's activity, sir, due to high amount of activity of microbes, there has been an increase in animals up to 60 days, sir, because the rate of decomposition of these residual application was much increased up to 60, sir, due to the availability of microbial activity of 60 days, sir. Subsequently, it has been decreased up to 90 days, sir. This then was seen similar in case of all the parameter stages in 30 and 60 and 90 in case
Ankala,
of 2 years
please
of.
conclude.
Yes, sir. Yes.
Your time is over.
Yes, sir. With all these parameters, sir, as you can see, this is a treatment showing that the green gram showed higher values, sir, compared with the soybean, sir. And this is my bacterial count, sir. Based upon all these parameters, I would like to conclude that, sir, the green gram with 100% albumin showing its significant effect of for the soybean groundnut, sir. With the change in the bacterial count, sir, there has been increase in the 15% increase from the past year, sir, in case of first year. Subsequently, it has been increased up to 25% in case of second year, sir. This shows that higher amount of biomass that has been added to soil surface will somehow increase the amount of bacterial count. It can be bacteria, fungi, or the activity of the urea, sir. However, the humus fashion was not much created, sir, but there's a positive advancement within the 2 years of study, sir. So overall, in case of Telangana, sir, we are taking up, there has been a crop diversification, sir, which we found one of the alternative. Based on this, sir, we are going for the legume-based cropping system, sir. So in case of Telangana also, there has been high amount of residues and high amount of biomass generated, sir, in case of green gram. So higher yields are seen in case of maize, sir. This improved both with the yields also, sir, and even the soil microbial activity also is also much more increased, sir. These are my references that I have taken, sir. This is my PPT, sir. Thank you so much, sir.
Okay, thank you, Ankala. So, any question from audience? Okay, if no question, thank you
Thank you,
to
sir.
Ankala. So, next presentation is from Ramaya KR. So, Ramaya, please share here. So, Ramaya KR, are you online? Huh? No.
रम्या कुछ नहीं ना ऐसे और कोई नहीं कोई है
और कोई नहीं
नहीं सर
Now we have reached the last part of the session. So there were eight presentations. So out of that, 6 are offline and 2 are online. So there are very nice presentation by different speakers. So first one is from Centurion University, who presented on the amaranthus and the lantana camera, their allelopathic effect on the horse gram. The second was the micropropagation of limonium, that is mainly based on the tissue culture. And the third one is the bacterial endophytes of wild rice and their potential for rice plant growth promotion and showing defense response toward the rice diseases. And the fourth one was the accelerated rejuvenation of paddy field through microbial interventions. That was from Maulana Abul Kalam Azad University, Kolkata, West Bengal. Then another in the next, Dr. D.S. Gujjar. So he has presented about the wastewater and their application in the turf grass. That was from WTC IARI campus. And the last one in the physical presentation was assessment of phenotypic variability under normal light and low light stress condition in diverse rice genotypes. That is again from NRRI Cuttack. Swagatika Das was the presenter. And there are 2 online presentation. One is from IRI Prabhanjan Rane, She talked about the penological studies of the different grape species for ascertaining their utilization in grape rootstock breeding. And the last presentation was impact of legume residues and nitrogen level on soil biological dynamics in soil zero-till maize cropping system from Ankala Sai Kishore from PJTSU, Hyderabad. So these are the presentations from this session. So thanks to all the presenters for their nice presentations. So anything to say from any audience, any questions? If not, we can conclude here. So thank you all the presenters. Here we will conclude the session. Thank you all.
Thank you, sir.